Hello everyone. In this segment, I will discuss today about ethnopharmacology and natural products. So I'll start with the introduction. Ethnopharmacology is a highly diversified approach for drug discovery which involves the observation, description, and experimental investigation of indigenous drugs and their biologic activities, that is based on botany, chemistry, biochemistry, pharmacology, and other disciplines like anthropology, archaeology, history, and linguistics. These all are contributing to the discovery of natural products with biologic activity. Now, let's discuss about history and background of ethnopharmacology. Ethnopharmacology was initiated by the missionaries in the colonies interested in the use of pharmacologically active plants like the Jesuits in 16th century in Latin America, Louis Levin, Carl Hartwig, Alexander Strach, Richard Evans. These all people established molecular ethnopharmacology and they contributed a lot towards this particular domain. Robert Gordon Wasson and Albert Houghton, resulting in the isolation of psilocybin from psychoactive mushrooms, discovery of pharmacological principles in foxglove, that is digitalis, Cuvier first antimalarial quinine from cinchona. Similarly, aspirin was developed based on ethnopharmacological studies with the bark of the willow tree, which has been used traditionally in Europe to treat fever and inflammation. Recent example of model research in ethnopharmacology is the work on the Chinese antimalarial plant Artemisia annua, resulting in the recent development of artemisinin into new clinical semisynthetic antimalarial agent artemether. Let's talk about the contribution to the modern medicine through ethnopharmacology. So natural products have been the source of active ingredients of many medicine from 1981 to 2007. Almost half of the drugs approved since 1994 are based on natural products. 13 natural product related drugs were approved from 2005 to 2007. 60% of the anti-cancer drugs and 75% of the anti-infectious disease drugs approved from in between 1981 to 2002 could be traced to natural origins. In continuation, I'd like to say drugs like aspirin, atropine, artemisinin, colchicine, digoxin, ephedrine, morphine, cyclostigmine, pilocarpine, quinine, quinidine, reserpine, taxol, tubocurarine, vincristine and vinblastine are a few important examples of drugs developed from medicinal plants. For example, atropa obtained from Atropa belladine, digoxin obtained from digitalis, ephedrine from ephedra stem, morphine from poppy seeds, quinine from cinchona, quinidine as well from cinchona, from Rauwolfia serpentina we can get reserpine, from Taxus brevifolia we can get taxol, vincristine, vinblastine is obtained from vinca alkaloids, that is Cathananthus roseus. So many plant-derived drugs are originally discovered through the study of traditional cures and folk knowledge. Some of these are still not substituted despite the enormous advancement in synthetic chemistry. Numerous molecules have come out of Ayurvedic experimental base, including Raufi alkaloids for hypertension, pisorolens for vitiligo, Flourina alkaloids in amoebiasis, glucosterols as hypolipidemic agents, picrosides for hepatic protection, phyllanthines as antivirals, curcumins for inflammation, with anolides and many other steroidal lactones and their glycosides as immunomodulators. So the moment we are using or we are taking help from ethnopharmacology, so we have to know what is the need of ethnopharmacology, why we are using all those. So here is the answer. The drug discovery process has become very complex and capital intensive, and companies with late discovery as a greater bottleneck. The usual distinctions between breakthrough and me too drugs may not be very meaningful, and today the pharmaceutical industry is technology intensive, not as innovative due to the shortage of new lead structure, with the strict regulatory processes adding more years to the discovery cycle and increasing cost due to the post-approval or post-marketing withdrawals. Random screening of plants used traditionally by pharmaceutical industries to discover new leads or drugs which are expensive and time consuming, but the ethno directed approach to traditional knowledge has been extremely useful in case of screening and identification of bioactive compounds with valuable applications in drug development. This has significantly increased the chances of discovery of new biomolecules with potential therapeutic application, while reduction in the cost and time involved in this process. There is also increased demand due to problems with drugs, resistant microorganisms, side effects of modern drugs, and emerging diseases with no available treatment knowledge of the indigenous people can give new resources. The ethnopharmacology knowledge, its holistic approach supported by experimental base, can serve as an innovative and powerful discovery engine for newer, safer, and affordable medicines for coming future. Now, I will discuss about ethnopharmacology and natural product drug discovery. So, ethnopharmacology and natural product drug discovery remains a significant hope in the current target-rich lead poor scenario. Therefore, several approaches towards the value of natural products are mentioned below. First, I will start with activity profiling of extracts, which says this kind of involvements screening mixtures of compounds obtained from plant extracts or from microbial broths but primary screens may not always contain novel compounds with the expected biological activity. Moving towards the next segment, that is biology oriented synthesis, where it says about it is kind of natural products act as a starting point for the search of active agents in new medicines. In order to match the natural material to its corresponding enzyme, scientists mimic nature by either introducing chemical residues into areas of biological relevance. Now, technology to measure gene expression. The proteome and the metabolome is available, which helps us better understand the mode of action of new compounds by comparing the changes in the transcriptome, proteome, and metabolome patterns to those observed with known drugs. Such an approach is now known as a systems biology approach. Now, moving towards metabolomics. These are the goal of metabolomics is to qualitatively and quantitatively analyze all metabolites in an organism. Currently, the methods that are being used in metabolomics can basically be classified into three different groups. The first one is chromatography based methods like gas chromatography and high performance liquid chromatography. Then moving towards molecular weight based method, which is mass spectroscopy. Next, moving towards the physical characteristics based methods, which are nuclear magnetic resonance spectroscopy, that is NMR. Now, problems are there along with the pharmacology. So, I would like to discuss about the different problems arises throughout the process of ethnopharmacological research. Many traditional preparations have been used for years and claimed to be the most potent and effective dosage forms, but very few scientific studies are carried out on these products due to lack of communication amongst traditional healers, physicians, and scientists. This gap can be filled by translating the old concepts in modern understanding, providing the possible explanation and hypothesis. Another problem for ethnopharmacology is the intense competition in drug research of medical field. There is requirement of stringent criteria to avoid development of false positive results. Malpractice in scientific method of ethnopharmacology is based on carelessly carried out bioassays using high concentrations of an extract or a natural product to demonstrate a pharmacological response. The general requirement for new drugs worldwide is quality, safety, and efficacy, but traditional medicine does not have double-blind studies or even well-described case records. Results of treatment of anecdotal observations in single patient cannot be submitted to health authorities anywhere in the world, and in most cases sufficient quality control and drug standardization is lacking for traditional recipes. Pharmaceutical biopiracy is a term used generally to describe the legal practice by pharmaceutical companies exploiting the indigenous people's traditional knowledge of medicine. The protection of intellectual property on traditional medicines should also be based on the guidelines for the protection of intellectual property on traditional medicines to be developed by the World Intellectual Property Organization, that is WIPO, with the treatment guidelines of such incidents and evidence-based evaluation methodologies along with the guidelines for ethnoarnogenics. A botanical drug or a preparation thereof is now regarded as one active substance in its entirely, whether or not the constituents with therapeutic activity are known. This is a major step in the development of new generation standardized botanical medicines. The WHO has published official documents on medicinal plants and WHO monographs on selected medicinal plants. Ethnopharmacology is a holistic approach to drug development using the latest technology for measuring as many different parameters as possible to discover possible leads to the mode of action, with ethnobotanists, ethnopharmacologists, physicians, phytochemists playing a key and vital role. It has already contributed to the development of modern medicine and is likely to play a more significant role in the years to come. The World Health Organization, that is WHO, has been active in creating guidelines and standards of botanical medicines. This will have great impact on future drug development and will prevent the loss of knowledge of traditional medicines and the loss of natural resources used as traditional medicines, particularly if some mechanism of loyalty comes into practice. Reverse pharmacology. Now, the last topic for this presentation is reverse pharmacology. So I'll start about reverse pharmacology, but I want to say something. There is a difference between pharmacology and reverse pharmacology. As the presentation will goes on, you will understand the entire thing. So starting with the introduction, traditional medicines emerged as a boon for the populations with strong socio-cultural and historical influences, or in the absence of alternative or complementary therapies. Ayurveda, an Indian system of medicine, built remarkable knowledge over the practice of several thousands of years. This gold mine of clinical observations is attracting global pharmaceutical corporations to fuel their investigational drug pipelines. Now we'll discuss reverse pharmacology, traditional medicine inspired drug discovery strategy. When a curious group of Indian scientists on trekking was introduced to the medicinal plant by a local tribe, an extraordinary story of a drug called Jivani began. This group of scientists was led by Dr. Palpu Pushpagadan, those who isolated the active ingredients responsible for energy boosting activity from Trichopas zelenicus. Reverse pharmacology is a rigorous scientific approach of integrating clinical observations, transforming experimental observation into leads by transdisciplinary exploratory studies, and developing leads into drug candidates or formulations through robust preclinical and clinical research. There has been several success stories of drugs developed from medicinal plants prior to the proposed organized approach of RP, that is reverse pharmacology. But the history makes it obvious how much opportunity loss occurred because of the time lag between the traditional use and the modern drug. The glaring examples are Artemisia, Cinchona, and Ephedra, etc. Sometimes the prevalent prejudice, cultural arrogance, or ignorance prevented any attention to the systems aligned to one's world views. The other factor was non-inclusion of the knowledge and traditions of other nations into the organized libraries and databases. Rauwolfia serpentina was demonstrated as an antihypertensive drug clinically as well as experimentally by Sen and Bose in 1931 at Kolkata, but the world did not take any notice of it until my teacher, means our ancestors, Vakil, 1949, published a paper in the British Heart Journal. The clinical side effects of Rauwolfia serpentina were meticulously reported by Sen, like depression, Parkinsonism, and gynecomastia, etc. As Kuhn has said, discovery commences with the awareness of anomaly, that is, with the recognition that nature has somehow violated the paradigm induced expectations that govern normal science. The watershed in new drugs for depression, Parkinson's disease, and galactoria emerged several decades later from the anomalies observed by Dr. Shen. Many more plants which show clinical effects need to be purchased systematically and in a time-bound manner by reverse pharmacology. Bedside observations on the established drugs may also have novel responses to consider. The path of reverse pharmacology can be applied effectively to the studies on the new indications for old drugs. The tragedy with thalidomide-induced phocomelia had alarmed the world and led to stringent regulatory requirements. But the discovery of a safe use of the optical isomer in multiple myeloma has been a great advance. Several such examples are known in the medical literature. Botulinum toxin and spasmodic tococolis, hydroxyurea for the induction of fetal hemoglobin, aspirin for platelet aggregation inhibition, and vitamin K27 for muscle cramps. The constant vigilance at the bedside may offer many more such opportunities for the enlargement of the indications of known drugs. The stigma attached to use of known drugs for unauthorized indications can be overcome in a fast track by the application of reverse pharmacology to the problem. What are the future scope? The current paradigm of drug discovery is shaken by the failure rates of the drug candidates at different stages of development. The long-standing traditional wisdom of Ayurveda can be explored for drug discovery and development. The experimental data can be explored further by relevant in vivo and in vitro experiments. This transdisciplinary approach of reverse pharmacology has sizable scope for new natural drugs and their derivatives. Efforts in this direction are already showing promise. The transdiscipline of reverse pharmacology needs a regulatory recognition and support for its development. We have to relook at the novel human biodynamic effects and pursue these for their therapeutic potential. Asia, Africa can offer reverse pharmacology for a better integrated healthcare for future. Thank you so much.